All right, good afternoon. I'm going to go ahead and get us kicked off. My name is Justin Jordan, and I'm a neurologist in Boston, and I'm here today really happily representing the American Academy of Neurology and the American Brain Coalition. Today, we're here to talk with you about the Brain Research Through Advancing Innovative Neurotechnologies initiative. This initiative is generously funded through congressional appropriations as well as funds from the 21st Century CARES Act, and is incredibly important work that's advancing our understanding of the body's most important and complex organ, the brain. Since its inception, the Brain Initiative has invested over $3 billion in more than 1,300 research projects and really engages not only scientists from many areas of neuroscience expertise, but as well includes mathematicians, engineers, physicians, and various other interdisciplinary teams. By accelerating the development and application of innovative technologies, researchers are able to show how individual cells and complex neural circuits in the brain interact with both time and space. This is laying the groundwork for developing new treatments for brain-related afflictions and beyond. And I'm really glad to be with you here today to talk more about this, this important initiative. If we could please advance our slide. I'd like to just start by certainly acknowledging our appreciation for the Congressional Neuroscience Caucus, specifically Representative Lubinauer and Representative McMorris-Rogers for their support today and for letting us provide this briefing for you. And now I'd like to introduce you to Dr. John Nye, who is the director of the NIH's Brain Initiative, the Brain Research Through Advancing Innovative Neurotechnologies Initiative. Prior to joining the Brain Initiative, Dr. Nye was a faculty member at UC Berkeley for over 25 years, where he served as the director of Berkeley's Neuroscience Graduate Program and Helen Wills Neuroscience Institute. An accomplished scientist who uses cutting-edge approaches to study brain function and neural regeneration, his work has led to the publication of more than 80 scientific articles in some of the field's most prestigious journals and 10 US and international patents. The Brain Initiative was announced in 2013, with Dr. Nye joining as its first director in 2020. Prior to his role as director, Dr. Nye served as the previous co-chair of the NIH Brain Initiative Cell Census Consortium Steering Group. And as director, Dr. Nye oversees the long-term strategy and day-to-day operations of the NIH Brain Initiative as it strives to revolutionize an understanding of the brain in both health and disease. Throughout our conversations today, we welcome questions through the Q&A feature at the bottom of your screen. And now if you could please join me in welcoming Dr. John Nye.
Thank you, Dr. Jordan. Can you see my slides?
Yes.
Terrific. And many thanks as well to the American Academy of Neurology, the American Brain Coalition, and the Congressional Neuroscience Caucus for this opportunity to highlight the unique role that the Brain Initiative is playing in developing critically needed tools and resources to accelerate the pace of discovery that ultimately, we believe, will lead to new cures sooner for devastating human brain disorders. I'd like to start with a personal story. So it's about almost exactly 3 years ago in the fall, in the before times, my wife and I were attending the wedding of a really good family friend's son. It was a great occasion. It brought together many families, that some of whom we hadn't seen for a while. And sitting at dinner, I struck up a conversation with an old friend, Deborah, a wonderful artist and painter. I hadn't seen her for a while, so it was a great chance to catch up. Our daughters grew up together. And she had asked me what I'd been up to. And this was just when I decided to leave this great life as a Berkeley professor to join the NIH, to lead the NIH Brain Initiative. So I told her with great excitement what I was up to, all the great possibilities for the things we're doing, what a great time it was and is for the field to bring in all these different disciplines beyond biology, math, physics, chemistry, engineering, to bear on how this most complex organ in the body works, the brain, and how it doesn't work when it's broken. And Deborah's a very smart person, very intense person, and she pushed me harder. She locked my gaze with her eyes. She said, no, tell me more. Be specific about how this is going to help people with neurological disorders. And she told me she had a vested interest. And at that point, I realized the whole time we were sitting there, she had her hands in her lap. and I looked down and she was suppressing a tremor. Deborah had Parkinson's. So for my friend Deborah, for many of you with friends, families, loved ones with these degenerative diseases like Parkinson's, Alzheimer's, ALS, as well as other disorders, neuropsychiatric disorders, which are devastating, substance use disorders, the clock is ticking. A cure cannot come fast enough. So what I hope to convey to you today is that through accelerating the pace of discovery about how the brain works in health, what goes wrong when it doesn't work, the resources we're developing here will help accelerate the search for cures sooner rather than later. The clock is ticking. So, the mission of the brain research through advancing innovative neurotechnologies is to revolutionize our understanding of the human brain by accelerating the development and application of new, innovative, and frankly, very cool technologies. This was announced by the White House in 2013, with the first awards being made in 2014, and represents a partnership between 5 US federal agencies and private foundations. The NIH efforts have been guided by 2 prescient and incisive strategic plans, the Brain 2025 report, which was issued in 2014, and an update on these strategic plans in the Brain 2.0 reports in 2019. The NIH Brain Initiative involves 10 of the 27 NIH institutes and centers, and our goal here is to develop and apply new tools for understanding how neural circuits underlie complex behaviors in health and how they go wrong in disease as a way of leading toward future cures. In order to achieve this goal, we will be leveraging emerging technologies to enable new discoveries about how these circuits function. We aim to use these discoveries as a foundation for developing new therapies for human brain disorders. And very, very importantly, we must disseminate and democratize these technologies for both basic discovery as well as for clinical applications for the benefit of all. Let me just take a minute and go through Brain by the numbers. Dr. Jordan mentioned that we have invested by now over $3 billion with a B dollars in over 1,300 brain projects since the first projects were funded in 2014. This has been an incredible investment, and I think we're already seeing dividends. Just by the numbers, our brain-funded investigators have been remarkably productive, leading to the publication as of end of 2021 of over 4,600 publications. Now the number is well north of 5,000 publications. You'll see that we've been publishing, our folks have been publishing in a wide variety of journals, reflecting the truly interdisciplinary nature of the work we're supporting. And this really does reflect the need for having multiple disciplines and expertise to come to bear on this set of hugely complex problems. So I would like to leave you with 3 key takeaways at the end of today. The first is that we're already seeing advances in the clinic from brain-funded projects. So I think we're already seeing dividends being paid out on this remarkable investment. At the same time, we're laying down a foundation to bring future cures and more advanced therapies to us on a faster timeline. So short-term, medium to long-term. And then in order to accomplish these goals, we're changing the way we're doing science. And I'll be talking about that toward the end of my presentation. So let's focus on this first takeaway. But people often ask me, you're investing all these funds, you're leveraging all these projects in these really cool areas, developing technology, better tools for understanding the brain. What's the payoff? Are we actually helping human beings today? And the answer is, yes, we're starting to. Let me just give you one example, just one example. It's a very exciting time for deep brain stimulation therapies. Many of you are probably aware that deep brain stimulation, or DBS, has been around for decades, pioneered in the treatment of the symptoms of movement disorders, in particular in Parkinson's disease. Now, we've been making advances in developing better DBS stimulators for Parkinson's. But in the meantime, there are other circuit disorders, we call them, disorders of the circuits in the brain that drive behavior, that also need attending to. And some of these are even more complex than we see in Parkinson's disease, where the locus that's being affected is quite well known. And one of these is in mood disorders. For example, in treatment-resistant or treatment-refractory depression, every patient is going to be a little bit different. depression is not one thing. It actually encompasses a number of different conditions that, again, are very particular for the person. And early efforts at using DBS to help treat the symptoms of depression initially showed great success, but then started kind of falling by the wayside a bit because of lack of success. Well, it turns out we really do need to appreciate the individual variability, the variation in people. And so here is the study from Dr. Samir Sheth at Baylor College of Medicine, where they tailored this DBS therapy to the patient. So in the upper left here, you see this, this group of surgeons and investigators looking at a holographic display of the circuitry that they suspected might be involved in this patient's major depressive disorder. They tailored the placement of both recording as well as stimulating electrodes to this particular patient. And then what was really cool here is they monitored that patient for over a week. And they recorded neural activity. They took both objective as well as subjective assessments of the patient's mood. Was the patient in a good state or a bad state? And they were able to correlate these activity patterns, these so-called activity biomarkers, with that patient's mood. And then that through very targeted stimulation in key areas, they were able to actually shift that patient into remission from that patient's poor mood state. So this is remarkable. This was a concept that came about maybe 8 or 10 years ago, frankly, was science fiction. And now we're seeing in this case study, there's another set of case studies from a group at UCSF that are showing that these personalized or even adaptive DBS stimulation protocols can be used to treat in a personalized way severe mood disorders, for example, treatment-resistant depression. So this is actually quite exciting, and now we're starting to roll this out into larger clinical trials. Hopefully we can disseminate this out more broadly to treat people who are really suffering debilitating mood disorders. So this is just one early payout of what we're seeing from technologies that are being fueled by the Brain Initiative. And again, 5-10 years ago, this to me at least was unimaginable. I'd like to talk to you a bit about what we're doing to lay down a foundation or a longer runway for identifying future cures. Much of what I just described to you is treating symptoms. As we start talking about various complex disorders as well as degenerative disorders, what we really are looking for is not just to treat symptoms, but also to slow disease, ultimately to cure, and hopefully one day to prevent disease. In order to do that, we need more information about how the brain works in order to learn mechanism, which in turn will lead to cures. So in addition to these exciting new advances with existing technologies like DBS, we also have our sights set on developing novel therapies for brain diseases. If we know better about how the circuits work, we can intervene, perhaps even with molecular gene therapies. So one of the big challenges we have is one of scale. The human brain comprises around 86 billion with a B neurons, about twice as many non-neuronal cells, and they're all functioning in a circuit that gives rise to things like cognition, emotion, behavior, etc. And what we need to do is we need to understand how they work normally and how they go awry in these various, I mentioned neurodegenerative diseases, but also in other conditions, including, for example, which has come to the fore, conditions that involve neuroinflammation, which we think also includes things like long COVID. So in order to understand how this marvelous biological computer works, we need a parts list. We need a wiring diagram. If one wants to know how a computer works without that knowledge, it's going to be a very tough go. We also need a way to interrogate and modulate these circuits. So if the parts list and the wiring diagram gives us the ground truth for what the brain looks like, we then need a way of asking very specific and targeted hypotheses about how we think they may contribute to behaviors, and ultimately how we can manipulate them in models of disease, and ultimately, again, with the goal of developing more advanced, more sophisticated therapies for circuit disorders of the human brain. So this is really looking into the future, but laying down a solid foundation for us to get there. And I think given the progress we've made over the past 5 years or so in developing these so-called ground truth informations, part list and wire diagram, I think we really have a good shot at changing the field in a very, very positive way. Let me just review for you very quickly a foundation for that we've built for building a so-called parts list of the mammalian brain. So this effort derives from the Brain Initiative Cell Census Network. It was a consortium of many labs actually across the world. In 2021, this international collaboration led to the publication of a large number of articles, 17 articles in the journal Nature, as well as 10 other articles in Nature's sister journals. And it was really a testament to what team science can achieve. And what we did here is we analyzed a very tiny portion of the brain across multiple mammalian species, the primary motor cortex. This is the part of your brain that controls movement. And it was analyzed at unprecedented depth and breadth using cutting edge, bleeding edge techniques across multiple domains. And it gave for us the organizational principles underlying the diversification of brain cell types in the mammalian brain. And right now, in the current state of the art, we can identify upwards of 3,000 to 4,000 distinct cell types that comprise these 86 billion total cells between your ears. Again, a testament for the power of team science. This is a remarkable effort that previously was really only seen in high energy physics and astronomy, where we really brought together hundreds of researchers across 3 continents from 45 institutions to work together to give us a great product that could not have been possible by any one lab alone. So what's it doing for us? Well, already it's paying dividends. Using this atlas, this parts list, I call it a treasure map of cell types in the brain, in the mammalian brain, including in the human brain, together with their molecular characteristics, their physiological characteristics, their connectivity. Already these tools have been applied in 2 separate studies to identify the cell types that are specifically lost in Alzheimer's disease patients, as well as in patients suffering from Parkinson's disease. So by identifying the cell types that are lost and having molecular information about them, knowing their properties, this will give us critical information about mechanism for why they might be lost. And therefore, with this information about mechanism, it gives us some insights about where the next set of where the cures might come from and how we could even prevent this degeneration. So this is really, really exciting stuff. These studies also lay the groundwork for the Human Brain Cell Atlas, which is going to be a huge effort, and it's an effort we're starting right now. This is just a figure from a preprint that was just released this week or end of last week from a group funded by the Brain Initiative in part, starting to describe the cell types across the entire human brain. And there's going to be a remarkable set of information coming at us. Really, this is an exciting time. I find it hard to express my excitement, how far we've come. I would not have believed 10 years ago, even 5 years ago, that we'd have this capability now to accomplish a full human brain cell atlas at this depth within the next 5 years. So this kind of gets me to the point of we're changing the way that we're doing science in order to accelerate the pace of discovery. So the Brain Cell Census Project has led in part to our standing up of what we're calling the Brain 2.0 transformative projects. So as I just mentioned a few moments ago, a deeper mechanistic understanding of the circuit basis of complex behaviors requires a ground truth knowledge of the components comprising circuits and their patterns of connectivity in the brain. And we also need ways of testing hypotheses for how these components work, ultimately leading to new therapeutic modalities, including, I hope, one day gene therapies for human brain disorders. So inspired by one of our strategic plans, the Brain 2.0 Working Group plan, was this idea of standing up now at a good time, some focused projects that could truly transform the way we conduct neuroscience research, ultimately with the aim of finding new cures. And this is a great time, not only due to the progress made by the Brain Initiative itself, but across the field of neuroscience in many, many adjacent domains in engineering, chemistry, physics, mathematics, data science. This is just a great time to think about standing up these large projects. So there are 3 of them that we've decided to move ahead with. They're all now fully launched, and they're deeply interlocking. And the first at the top here is a comprehensive brain cell atlas with an emphasis on the human brain, a very, very large challenge, but over the last 5 years, we've really developed the technologies that allow us to do that. The second here on the lower left is the brain, whole brain microconnectivity project. So basically developing a wiring diagram. What are the trillions of connections that are made between different areas and different cells in the brain that give rise to these circuits that in turn give rise to these complex behaviors that we observe. And then finally, in the lower right, tools for gaining precision access to brain cell types that will allow us to use this ground truth information in order to test hypotheses with greater precision in a virtuous cycle that again will lead to better hypotheses, a better understanding of how neural circuits underlie behavior in both health and disease, and again, ultimately leading to better circuit therapy. So we're very excited about this. This will be all in service of better understanding how the brain functions, again, in both models of health as well as in different disease states. So this is one way that we think we will be changing the face of neuroscience by promoting these large team-based efforts to enable hypothesis-driven research that will lead to deeper understanding for how this incredible biological computer works. Now, much of what brain does involves humans. We have a series of small first-in-human clinical trials, as well as opportunities to study how neural circuits function in human subjects that are undergoing treatment for other disorders. And so it's very, very important for us to be highly aware of and anticipatory of any issues that might arise due to the ethical, legal, societal implications of this work, in addition to the paramount concerns about patient safety and privacy. But since we are intervening into the brain, which defines who we are, we're also quite concerned about issues of agency. If we help a patient, for example, suffering from severe depression or for substance use disorder, by treating that person's symptoms or even curing them of that symptoms, are we changing who that person is? And does that person really fully understand what that means? Are their families fully cognizant of what that means? Treatments that we can develop to alleviate symptoms from disease might also be used for other purposes, for example, enhancement, or even in malign use. So in this regard, we have a variety of mechanisms and groups that help us study these issues in a proactive way. Very, very important is to engage the public in these discussions, and so that we have full buy-in and guidance for what will be the right thing to do, or certainly to avoid the wrong things to do, and ultimately to democratize these technologies, these resources, these riches, again, for the benefit of all. Another key cornerstone of the BRAIN initiative is to advance scientific excellence in any way that we can. And one way is through inclusivity in our workforce. Now, there's a growing body of evidence that's giving a compelling story that teams with diverse backgrounds, perspectives, expertise tend to outperform teams that have a more homogeneous makeup in terms of innovation, creativity, and actual outcomes. And to this end, we're very, very interested and committed to leveraging all the talent that's available to come to bear on the most complex organ in the body, the brain. We certainly cannot afford to leave any talent on the table. So to this end, BRAIN has embarked on a number of strategic avenues. We have programs specifically targeted for broader workforce development here in the upper right. We have programs for capacity building at under-resourced institutions, and as well as dissemination of resources across the globe, as well as for compelling our researchers to think about how they can diversify their teams to bring better outcomes. So this is all a work in progress, but again, it's all toward the goal of doing better science and bringing cures to us faster. So again, I'll just summarize the three key takeaways I'm hoping to leave you with today. BRAIN-funded advances are already making impacts in the clinic, and we hope to expand that as the time goes on. We're laying down what I believe is a very strong foundation for the future, so we can move from treatments to actual cures for these devastating brain disorders. And in order to do so, we're creating a new paradigm, or a set of new paradigms for doing science, with a goal of doing better science and accelerating the pace of discovery. Of course, there's a fourth takeaway, right? And I'd like to convey to you that BRAIN is uniquely situated to pursue new cures, new cross-cutting opportunities in technology and resource development that are not and would not otherwise be being pursued at any one NIH institute or center, which is focused in a specific disease area. And I'd also like to emphasize that we're not just adding incrementally to the many great advances supported by NIH in the neuroscience space, but rather we're proving to be a strong force multiplier that is accelerating the pace of discovery by providing broadly applicable tools that promise to bring much needed cures on a shorter timeline. So it's critically important for us to continue building this momentum, to push the field forward, to make today's science fiction tomorrow's reality, and to have tomorrow come sooner rather than later. So I'll thank you very much for your attendance and your attention. You can learn more about the NIH Brain Initiative at our website here.
Dr. Nye, that was really spectacular. Thank you for the incredible overview. And I have to say, the advances that are coming out already are so impressive and obviously impact a variety of neurologic diseases. So we're going to open now for Q&A from the audience, and I'll encourage those of you who are attending to please put in questions into the Q&A, and I'll monitor it. I'm going to start with a question maybe from the start, if that's okay, Dr. Nye.
Yep.
So the science and technological advancements you highlighted sound both complex and pretty fascinating. What is it that makes the Brain Initiative special, though?
Yeah, so that's a great question, and as I mentioned at the very end of my talk, it sits in a unique space at NIH, and I think as well as in neuroscience. So each NIH institute or center is focused on a specific area. For example, National Institute on Mental Health is focused on mental health issues. How do we identify, characterize, and ultimately find ways to treat folks with mental health issues? National Institute of Neurological Diseases and Stroke, again, is focused on neurological diseases, and so on and so forth. But what's in common across the 10 brain participating ICs is neuroscience, the functioning of the nervous system, and the tools we build to look at one area could be very well applicable to others. So we're really kind of sitting in this unique space that's not covered by any one single disease-focused IC or institute or center. And by building a foundation of new tools that can be used across the board, I think that really is very special. And the sustained investment in brain allows us to go beyond a patchwork funding way. I mean, there are discoveries made across the board, don't get me wrong, right? There's many great things. And the idea here is to leverage and coordinate these things in a focused way. The NIH Brain right now comprises about 5% of the total neuroscience budget at NIH. And the idea here is not just to add incrementally to that, that'd be 5%, but really, again, to have an outsized impact so that we're not just doing piecemeal approaches, but we really have a coordinated way that has a basis, a biological basis for treatment and a mechanistic basis for treatment that can be applied across the board. So I think that's what makes us unique. And frankly, I think we're kind of special.
From my perspective, I think that came across really nicely in the slides that you presented, the broad reach that all of these have. So just looking at some of the other questions, in your opinion, Dr. Nye, in your 2 years at Brain, what has surprised you? How does that compare to what's been accomplished in the lifespan of the program?
Right. I mean, what surprised me, frankly, is just the pace of discovery, the pace of innovation. It's not just been within Brain, but it's really leveraging multiple revolutions adjacent and partially overlapping with what we're doing. So there have been multiple revolutions. One has clearly been in high throughput DNA sequencing. So there's been the genome revolution. There's been a revolution in material science, computer science, data science that give us better materials with which to build electrodes that allow us to record more finely from more and more cells at a time. There's been the gene editing revolution, right, which is just only a decade old, and the pace has been breathtaking. Again, I mean, we talk about Moore's law in the electronics industry, and I look back at the work we're doing now. For example, just in the cell census project, back when we started this project in 2014, I was actually involved. If you published a paper with 1,000 cells, well characterized, these are just numbers. You were bleeding edge. You were state of the art. The work that I just showed you about involves over 3 million cells and counting. So we're talking about 3 orders of magnitude improvement in scale, which is not just scale. With the scale gives us information that we never otherwise could have gotten, right? So there's a point at which you don't just get more. By getting more, you get different. And now we're getting 3 orders of magnitude different. To me, achieving that in the last 8 years is frankly mind-blowing, for lack of a better term. So the pace of discovery, but not just here, our ability to record from thousands of neurons right now in the brain simultaneously was a dream. Actually, it was an aspirational dream that was talked about, published about 10 years ago. Wouldn't it be great if, and now we can do it. And so the beauty there is by being able to record from multiple parts of the brain at once, we're now able to really understand how different parts of the brain are engaged in generating behaviors. How the different parts of the brain engage and how do they change when we change our moods or change our states. The analogy I like to draw, it's not my analogy, is that if you want to fully appreciate a symphony, you want to hear every instrument in the orchestra, not just the principal violinist, not just the oboist, right? So this really gives us the ability to really get in deep and understand the complex inner workings. So the pace of discovery is what's really been, to me, a surprise. Maybe I shouldn't be surprised, but it's really been quite amazing.
Yeah, absolutely. And obviously that single cell sequencing and the genetic alterations are finding their way into various other aspects of medicine well beyond brain related research.
Absolutely. And it's been mutually reinforcing too. I mean, there's a large international effort in mapping every cell type in the human body, the Human Cell Atlas Project, and there's been a lot of synergy here. So it's been, again, a great force multiplier. We benefit from the field, but many of the tools that are being used in the Human Cell Atlas Project and in applications in disease, including in oncology and what have you, many of these techniques were pioneered from brain-funded projects.
Amazing. So you gave us a great list, but how about, can you name what you think the biggest success of the BRAIN Initiative has been so far?
Right. I mean, clearly you can tell I'm quite enthusiastic about the cell atlasing project for a couple of reasons. One, it's amazing. I was, in full disclosure, I was involved in the early phases of that project. So for me, it's been a wonderful to see both up close as well as from my current perch. I think these and other large projects they are revolutionizing the way we do science. The big dividends are yet to come, but I really, from what we've seen so far, I think the future does look very bright. So that's one huge win. Also being able to see the advances in the clinic, right? I mean, we all know somebody that is just in desperate need for at least some relief, if not while they're waiting for a cure. Hopefully the cure will come in their lifetimes, our lifetimes. And being able to see folks looking at existing technologies, taking best-in-class technologies and just pushing the envelope that much further, I think to me is quite gratifying. And frankly, these first-in-human trials, they're expensive. They're limited in reach and scope. But to this extent, I'm a bit of a Pollyanna. I think this is a great start. We have been able to economize, democratize these, what used to be very, very complex, expensive treatments more broadly with creativity, talent, and the focus, the will to do it. So I'm very, very hopeful that in the meantime, while we're waiting for these longer term gains, that we can actually see that we actually are seeing gains here and now today.
Right. Yeah, that makes a lot of sense to me. I'm just going to remind the audience to go ahead and be sure you put in questions into the Q&A, and we're working through all of them here. So Dr. Nye, that was, I like that. I'm curious, what do you hope to see produced by the BRAIN Initiative as it continues to grow?
Yeah, so again, one of the starting out goals of the BRAIN Initiative is to really ask, how does the brain underlie complex behavior? How does it underlie cognition? I mean, you can think about the so-called simple things like what's involved in motor control, sensory perception. That's kind of in my field, my particular scientific interest. Cognition, emotion, right? But then we can get into kind of the more complex, well, even more complex issues, the issues of consciousness, which, people, including philosophers, have been been grappling with for thousands of years, right? What we're beginning to see is the ability to record from now thousands of neurons at a time, simultaneously from many, many different areas of the brain, to some extent to be able to do this in humans. Now we can start asking, well, what might be the nature of internal states, right? And so at the 25,000 foot levels, I really do see some major advances coming from these techniques because we have developed better ways of monitoring activity in these neural circuits. And it's a huge dump of data. So very, very importantly, in order to deal with those data sets, first of all, you need a model to start out with. You need a theoretical framework, which is being developed by many projects. And you also need a way of dealing with the data. So again, this is where data science, computer science critically comes in to help us not only interpret, but to design the experiments in a way that they become interpretable. So I think we're going to see a lot of really cool things over the next 5 years there. And again, for me, this reveals the inherent beauty of the brain. I mean, I'm a biologist. I just love looking at this stuff, right? But beyond that, if we understand some of these issues more in greater depth, then we can start asking what happens. For example, why is a patient suffering from depression that cannot be treated by any current therapies that we have available to us? Where might be the points of intervention, whether they be a circuit intervention where you know better where to place an electrode, what patterns of activity to use to stimulate it, or whether it's identifying a better drug that can be used. There's been a resurgence in the development of new antidepressants based on psychedelics, and Brain is also funding programs that are actually helping us identify better and more effective drugs with fewer of the side effects. So there's a lot of really great stuff coming that will enable the more precise targeting of these circuits in different ways with this basis of knowledge.
That's interesting. And, this is such a broad area that you're talking about here, but I'm curious, we had a question about what's the proportion of pediatric brain research compared to adults?
Right.
Can you line out that up for us?
Very, very, very important. So I'm a developmental biologist. I call myself a developmental biologist by training and certainly in my heart. How the brain is constructed and built is critically important for us for a number of reasons. One, it's just a fascinating problem. Many of the devastating neuropsychiatric diseases are developmental diseases. And even when we look at diseases of aging, neurodegeneration, for example, in Alzheimer's disease, we don't yet know the triggers for some of these diseases. But what we also don't know, but now we're starting to suspect, the triggers may start early. They might even start at the time of gestation because of some genetic or other environmental predisposition. Wow. So being able to understand how neural circuits arise, how they're constructed, what are the events that happen normally, what happens in diseases is critically important. We are making a conscious effort to do this in this very large Brain Initiative Cell Atlas project that we've just stood up. A critical component of that is looking at the human brain over the lifetime. So starting in gestational development through aging. And we believe this will give us some incredible clues about what's going on normally, which again, it's the beauty of the brain, I call it, but also give us insights into what might be happening when circuits are not being set up right. Many of the disorders we're looking at are disorders of circuits that either didn't get formed in a way that is within the window of what we call okayness, right? As well as other perturbations that might have happened because of environmental effects. And we really need to know what's going on in these critical periods, or the periods during development where we know that neural circuits form, certain behaviors are developed, and we need to really know what's going on there. And this is part of the one of the major goals of the Cell Census Project, is to understand across a lifespan, including in phases of early development.
Amazing. I think it's critical, I think, and I appreciate the attention to that. On the other spectrum, though, of course, the population is growing in age. And with this comes an increase in the prevalence of neurologic disease. So how do you see the cell, the individual cell research you're doing now through the Brain Initiative being able to reduce the burden of diseases, for instance?
Right. So again, we're not specifically focused on disease, but disease can tell us a lot about what's going on. So that's one piece of it. It really comes down to mechanism. If we look at Parkinson's disease, I mean, there's some familial forms that we have an idea what the drivers are. If we look at Alzheimer's disease, again, there are some familial forms of that, but really, by and large, we don't know what the drivers are, right? We don't know what's initiating the problem, and we don't know when. In order to get at ultimately cures, prevention, cures, treatments, we need to understand mechanism. So I think what the brain here can do is, we can invest for the long haul, but make the long haul be a bit of a shorter haul. I just saw a quote from my former colleague at Berkeley, Saul Perlmutter, who won the Nobel Prize in Physics a number of years ago. And the quote goes something along the lines of, the value of basic discovery is it allows one to leapfrog over big problems. And I think here is an opportunity to leapfrog over the big problem. The big problem we have right now is we don't understand mechanism driving the disease. So an example I can give is, I mentioned this briefly in my presentation, was one of our brain-funded investigators used techniques that were developed in the cell census network to identify in humans and characterize in humans the major cell types comprising the set of dopamine neurons in the midbrain. Now, some of these, this is where you have problems with neuronal loss and Parkinson's disease, which leads to these, devastating movement disorders and then ultimately in death. And what they did is they identified a population of cells that specifically are lost in Parkinson's disease patients, because what they did is they took these techniques and they applied them to a large number of samples that were derived from Parkinson's disease patients. So they actually could see, who's missing, right? Who's missing, who's lost. Okay, so that's fine. So we knew neurons are lost, but now they know what they look like, right? They know what genes they're expressing. We can get an idea about their character. We can now model this in non-human systems to really understand what they're about. What was really cool is they took that information and they looked at genetic information, population genetic information that identified genes that were mutated or had changes in sporadic form associated with sporadic forms of Parkinson's disease. And some of those genes that were identified as potential culprits in sporadic PD were actually expressed in these cells. So now you start getting closer to some smoking guns.
Absolutely.
Right. So, the issue with these degenerative diseases, what we're coming to appreciate more and more now, is the participation of these non-neuronal cells. So it's not just that neurons are dying all by themselves, but it's their interactions with other cells in the brain. There's these neuroimmune interactions that seem to be critically important in a spectrum of disorders, degeneration, in ASD, in autism spectrum disorder, in long COVID, right? And so by knowing who the players are, by knowing really who they are, not just it's cell type A, B, or C, but this is their characteristics, now we can start drilling down on mechanism. And once you know what makes those cells tick, and once you start getting an idea of why are they dying, then you've got a target. You have a set of targets that we can start drilling down on in terms of finding a way to prevent their loss and ultimately, independently to maybe replace these cells through other means, but certainly as a way of intervening early. So I think this is the investment for the long haul, and the hope is that by leapfrogging through with these basic discoveries, the long haul actually isn't such a long haul after all.
Right. No, that makes complete sense. I want to hit another point there, though. This is obviously Parkinson's and so forth, but we had a question about how does this work potentially interact with cancer, for instance? And can you talk about that a little bit?
Right. So, cancer per se is not directly within our remit. It's not one of our 10 brain ICs, but it's not to say that we wouldn't have an impact in the field. This is your field, right, as well. So, we're seeing a lot of really important work coming out about what are the cells, what are the origins of cells involved in different types of cancers of the brain. There's been some amazing work, in particular from Michelle Manji, looking at some of the driving factors that stimulate progression in glioblastoma and metastases and the like. And again, it comes down to knowing what you're working with, right? So the other week, the check engine light on my car came on. Actually came on months ago. I finally brought it into my mechanic, right? That's what he does. He plugs it in to his little machine and it tells him, well, where's the fault? And he goes and he knows the parts, all the parts and all the workings of the car, and he was able to fix it pretty quick, right? We need to know that here. We need to know what's going on. Not only what are the origins of some of these tumors, which is very, very important, what genes are they expressing that we might be able to find as druggable targets? And very, very importantly, for example, from Dr. Manji's work, what is the role of environmental effects, activity in stimulating these cells to do bad things, right? So it's really just having the basic tools for understanding how the nervous system works and then applying that to these different diseases to understand how to either ameliorate for now, but ultimately cure later their progression. So it's really about providing the general tools here across the board.
I love that analogy with the car. That's perfect. I think that makes a lot of sense.
It's a true story too.
You mentioned long COVID as well, and just curious if the Brain Initiative has had any engagement and looking specifically at long COVID.
Not directly, but again, I sound like a broken record. It does give us the insights in identifying the cell types that are involved. There were there's this interesting phenomenon as well of COVID-associated anosmia. Anosmia is the loss of the sense of smell. And in fact, one of the earliest biomarkers for COVID is the loss of smell, at least in the pre-Omicron era. And it became kind of a mystery as to, well, why? What is this telling us? And the other side of that question was this observation that's been around for decades that many patients suffering from neurodegenerative diseases, including PD, Parkinson's and Alzheimer's, they tend to lose their sense of smell. And loss of sense of smell is also associated with aging. And this is always fascinating me. Why? What's the connection between your inability to smell your coffee in the morning with these other complex and devastating degenerative diseases? Well, it turns out that just by applying it was by applying the analysis of existing single cell data sets, one of which I contributed to, we found out that the virus actually does not have a way of infecting the neurons that actually receive the smells, but actually the support cells. Okay, so this was a way of winnowing down what the possible hypotheses could be, right? So science is about eliminating possibilities in order to get at the actual thing. There's been a beautiful set of studies from Dr. Stavros Lomvardis at Columbia University, who's looking at the effects of SARS-CoV-2 infection on the loss of sense of smell. This might seem esoteric to you, but right, it turns out that we were only kind of part right when we said it doesn't involve neurons, which it doesn't. It probably also is independent of the other cells in the respiratory tract. It seems that the loss of sense of smell is due to some systemic response, some humoral neuroinflammation, inflammatory immune response that's affecting the sense of smell indirectly. Now, this could be a window into understanding mechanisms driving degeneration, right? So it's the body is highly complex. The more you know about mechanism, the more better tools you have for getting in there. You never know when you're going to get a surprise where, as Pasteur said, chance favors the prepared mind. You might be able to gain an insight into this mechanism. So the shorter version of this very long-winded answer is we're not targeting long COVID through brain directly, but we're providing the tools that allow people to understand it. So for example, there have been a number of projects that have very beautifully characterized the non-neuronal cells and the neuroimmune cells and cells of the vasculature of the brain, and how they differ in different conditions and how they differ depending on where they're located. And that's going to be information critical, I believe, for understanding the basis of long COVID, which from what I can tell from my 10th row view of the literature there, it's going to involve neuroinflammation. It's clearly involving neuroinflammation, and you got to know who the players are in order to sort that one out.
Yeah. Makes complete sense. Thanks for that. So I'm going to move on. The BRAIN Initiative works across disciplines and institutes to provide comprehensive research. How does the funding get prioritized throughout these varying arenas, and what does that mean for the future of the initiative?
Right. So we're kind of in this interesting space, right? I mean, I'm a basic scientist by training, in my heart, in my brain. I live for a great hypothesis-driven project that's going to ask a great question and get a great answer. But in order to ask better questions and get better answers, you need better tools. And here is perhaps a dichotomy, and I would argue perhaps a false dichotomy between the importance of individual, inspired, hypothesis-driven research versus these large science projects that are geared at generating resources. And I say it's a false dichotomy because they actually exist in a virtuous cycle, right? Biologist goes in the lab, thinks of an experiment, does an experiment, has a better question to ask, but geez, I don't have the tool I need to ask this. I need to record from more neurons simultaneously in the brain to do that. Necessity is the mother of invention. Somebody, that person or somebody they're collaborating with or somebody else coming in says, you know what, I've got a better material that I can make. I can make a finer electrode that does less damage to put it in the brain. In fact, maybe that electrode can be flexible and I can put more sites on that. And fast forward a couple of years and we have electrodes that can now record from hundreds, if not thousands of cells simultaneously in the brain. And now that biologist can go in and say, okay, I can ask my question with much greater precision. And the answers I get out of that with all the data and my ability to understand the data, I'm going to ask the next better question. And you see, you just keep on going around and around and around. So we have these large resource generating projects. I mean, the transformative projects that I mentioned, the cell census project, the connectivity project, this project to develop tools for interrogating neural circuits. These are large resource generating projects. They're far beyond the capabilities of any single lab if they're going to have the impact that we want it to have. And that's all in service of the folks who are interested in asking and answering specific questions. And some of those questions come about through smaller projects, and some of those questions come about through larger projects. So, it's not a one size fits all, but it's taking this coordinated and strategic approach to leverage the best mechanism, the best strategies to suit the problem at hand. And I think we've done a pretty good job of balancing that, but we're always trying to refine and adjust where that balance lies at any given time. Perfect.
So we've got just a little over 5 minutes left. If the audience has any residual questions, I'll ask you to put them in the Q&A. I have one more question for you, Dr. Nye. Brain health across the lifespan is a growing priority. You may know that the American Academy of Neurology hosted an inaugural Brain Health Summit in DC in September. How do you see the outcomes of the BRAIN Initiative improving brain health now and in the future?
Yeah, I mean, this is what's on our mind all the time. I hope I conveyed over the last 45, 50 minutes where we're positioning ourselves in that domain. And again, it's about taking what we have now and taking best-in-class therapies, treatments, technologies, and just squeezing a little bit more out of it. And sometimes you can cross the threshold and boom, you've got something that you didn't imagine a few years ago. Again, to me, the ability to treat mood disorders, other mental illnesses, OCD, substance use disorders, with DBS, which has been a technology around for over 20 years, is literally mind-blowing, is brain-blowing, right? I think that's very important. But again, I think it's really the longer-term investments, which I'm hoping it won't be too long, to bring the horizon closer to us. And I really do believe that with a greater mechanistic understanding of the brain and how the circuits work, how the cells participate in the circuits, this is the key to unlock the door.
Absolutely. I think that's tremendous. Thank you for those answers. I'm looking at the Q&A and I think we've answered everything there. If anybody else has any last minute questions, we'll add them. Otherwise, I think we're coming to the end. I would just like to say this has been a tremendous opportunity to learn more about the projects coming out of the Brain Initiative, and I'm fired up at the discoveries you've made already, and at the enormous opportunity for how these can impact my patients and even myself and my family in the future. So thank you so much for all of that. I think that we've answered all of our questions in the Q&A, Dr. Nye. Any last thoughts before we leave?
No, I just want to thank you first of all for being here, for facilitating. It's really inspiring for us always to be connected with folks treating patients. This is the source of our inspiration and our motivation. Again, I'd love to thank the AAN and ABC for organizing, the Congressional Neuroscience Caucus, our great friends, allies, and advocates on the Hill, and all of you here for your attention. We do look forward to continuing these conversations with any one of you. Brain Initiative, again, is playing an important role here in driving new technologies that will be critical for us to get at what makes the brain work. And we really are deeply invested. We do appreciate all the support here. And we are committed to making sure we pay dividends sooner rather than later.
Absolutely. Yes, thank you very much to everybody for coming and to the Congressional Neuroscience panel. All right. Thank you very much, Dr. Nye.
Thanks very much.